Hello everyone and welcome to today's Crush the MCAT lesson on proteins and protein formation. Let's talk first about protein structure. There are four layers or four ways to describe a protein structure. The primary structure of a protein is its string of amino acids. This is just the list of amino acids which make this protein, read from N-terminus to C-terminus. Important to note, all of these amino acids are held together by peptide bonds. The secondary structure of a protein is the local folding of the chain backbone. What that means is when you put the protein in a solution, hydrogen bonding occurs, which forms alpha helices and beta sheets. This begins to give the protein a sort of structure. The amino acid proline can cause beta turns in a protein, which can disrupt the local structure, especially in cases of protein mutations. Tertiary structure is the overall 3D shape of a single protein chain. This occurs when the protein is folded by non-covalent side chain interactions. Nonpolar or hydrophobic residues being another term for amino acids, will fold inwards away from water and towards themselves. Polar or hydrophilic residues will fold outward towards water. Salt bridges may form due to ionic interactions, especially with aspartate or glutamate. Two important things to note: cysteine residues will form disulfide bridges amongst each other, and all of this is driven by thermodynamic stability, meaning the final conformation of a protein in its tertiary structure is the most thermodynamically stable conformation it can have. Quaternary structure is when multiple protein chains or subunits join together to make one functional protein. Only some proteins have quaternary structure, and these are proteins made of multiple polypeptide chains. These chains can be held together by non-covalent bonds, like hydrogen bonding from polar residues, or covalent bonds, like the disulfide bonds we mentioned earlier from the cysteine residues. A classic example you'll see of this is hemoglobin, which has a quaternary structure because it's made of 4 subunits, versus myoglobin, which is a monomer without a quaternary structure because it's just made of one protein chain. Going through this again, the primary structure is the string of amino acids which make the protein. The secondary structure is the local folding of the chain backbone with beta sheets and alpha helices. The tertiary structure is the overall 3D shape of a single protein chain. And the quaternary structure is when multiple protein chains or subunits join together to make one functional protein. Speaking more about protein folding, the hydrophobic effect will cause nonpolar parts of a protein to face inwards. This happens because hydrophobic amino acids will cluster to minimize the disruption of water. This will increase entropy, which is thermodynamically favorable. Remember, the proteins fold to make the most thermodynamically favorable configuration, and when all of this occurs, water forms a solvation shell around exposed polar and charged residues. I have a diagram of this here. When you drop a protein into water, it begins as unfolded, which we can see on the right. And when this happens, water will have to make an ordered shell around each hydrophobic residue. Nature generally prefers entropy, which is disorder. So the water does not want to be ordered around each of these individual hydrophobic residues. Instead, it would prefer if all of these hydrophobic residues joined together and made one large hydrophobic clump. That way, water only needs to be ordered around one large hydrophobic clump, and then in the rest of the solution, the water is free to be as disordered as it wants to be. Again, the main point here is the hydrophobic effect increases entropy, which is favorable by nature, and that it will cause all of the hydrophobic residues to associate with themselves, and all of the hydrophilic or the water-loving residues to associate with the water freely. Denaturation is the unfolding of proteins. Unfolded proteins are inactive. This can occur with significant changes in pH or temperature. It can also occur with disruptive solutes like salt or urea or organic solvents. Chaperone proteins work to prevent denaturation and to refold unfolded proteins. A really important fact you should know for the MCAT: denaturation cannot affect the primary structure. Remember, the primary structure is just the list of amino acids read from N-terminus to C-terminus. No matter how a protein is folded, it can never affect that list. Post-translational modifications can further change proteins once they're translated. Ubiquitination can mark a protein for degradation by the proteasome. Glycosylation, which uses carbohydrates, marks a protein to be sent to the cell surface via the endoplasmic reticulum. This is common in secreted proteins and proteins made for the membrane. Phosphorylation regulates enzyme activity to make it active or inactive, only amino acids with OH groups can be phosphorylated, like serine, threonine, and tyrosine. Proteins can also undergo methylation and acetylation, and proteins can also go proteolytic cleavage to break proteins down in order to activate them, which you may see in the case of zymogens, or to inactivate proteins. Some transmembrane proteins you should know. G-protein coupled receptors, or GPCRs, transduce signals across a membrane using G-proteins and GTP. These receptors span the membrane 7 times. When a ligand binds to this receptor, G-alpha will exchange a GDP, or guanosine diphosphate, to a guanosine triphosphate. G-alpha acts as a signal until it hydrolyzes guanosine triphosphate back to GDP as an internal timer, and in the meantime what G-alpha will do is it will stimulate adenylocyclase, which creates CAMP, activates pKa, and then phosphorylates target proteins. GPCRs will use this to amplify signals, so at every stage here it gets exponentially bigger and bigger. That way, after just one ligand binding to one receptor, thousands of target proteins can be phosphorylated. Ion channels allow movement down electrochemical gradients. You'll see these being used for ions like sodium and calcium and potassium. Voltage-gated ion channels open in response to changes in membrane potential. Ligand-gated ion channels open in response to molecules or ligands binding. Pores do passive transport in the membrane. Aquaporin is a very important pore which moves water. Active transporters move molecules against a concentration gradient using energy. That can be in the form of ATP in primary active transporters, or it can be in the form of pre-existing ion gradients in secondary active transporters, which are usually set up by other primary active transporters. Receptor enzymes function as both receptors and enzymes. For example, the insulin receptor, which is a receptor tyrosine kinase, or RTK, autophosphorylates itself on tyrosine residues when insulin binds to it. So it can detect insulin binding and it can phosphorylate itself when that binding happens. So it's a receptor enzyme. Finally, you should be familiar with peptide hormones as a group of proteins which can bind to cell receptors to cause certain effects inside the cell. Here are some non-enzymatic proteins you should know. Actin forms microfilaments, and microfilaments support cell movement and cleavage like in cytokinesis and contraction. Tubulin forms microtubules, and microtubules provide intracellular transport tracts, like rails on a railroad. Microtubules are also important for eukaryotic cilia and flagella, which help move cells in solutions and have a 9+2 arrangement, which means there's 9 doublets of microtubules with 2 central microtubules. Centrioles, which are organized 9×3, which means 9 sets of microtubule triplets, organize the mitotic spindle during cell division. There's also intermediate filaments, like keratin, which provide tensile strength and stability. You should be familiar with collagen and elastin and keratin. These are major intracellular and extracellular support proteins. Antibodies, which are also proteins, bind specific antigens or ligands as part of the adaptive immune system. And finally, kinesins and dyneins are motor proteins which carry vesicles. These walk on microtubules and hydrolyze ATP. You should know the direction that these go, and I remember this by remembering kinesins carry out and dyneins dyne in. And that concludes today's lesson on proteins and protein formation. Thank you so much, and don't forget to like and subscribe.